And next, I have the honor of introducing our next speaker. So we will have Dr. Goodell and Dr. Rao presenting on what is DNMT3A, how does it work, and how does it cause TBRS. So Dr. Margaret Peggy Goodell is professor and chair of the Department of Molecular and Cellular Biology and director of the Stem Cells and Regenerative Medicine Center at Baylor College of Medicine in Houston, Texas. Dr. Goodell's research is focused on DNMT3A mechanisms in blood stem cells. Goodell was elected to the National Academy of Medicine in 2019 and is a recipient of many awards. And Goodell directs a laboratory of about 15 trainees. Also, Dr. Rachel Rao, who will be joining us virtually, is an associate professor of pediatrics at the University of Washington and a pediatric hematologist-oncologist at Seattle's Children's Hospital. Before her move to Seattle in the summer of 2023, she was a physician scientist faculty member at Baylor College of Medicine with Peggy and Texas Children's Hospital in Houston for nearly 11 years. Dr. Rao specializes in the care of children with blood disorders, including leukemia, and she conducts clinical research, and a major focus of the Rao Lab is the study of the effects on blood development of DNMT3A mutations, including those found in individuals with TBRS. So I'd like to welcome them to the stage.
Okay, I'm really excited to be here. It's been a really wonderful meeting so far, and I'm learning so much, and I'm really excited to tell you what we're thinking about DNMT3A. So I kind of wanted to start from the beginning and talk to you about what DNMT3A does, but I'm going to use an analogy to break it down, and hopefully it's not too simplistic, and hopefully it'll be interesting, and we can talk about more complicated stuff like after the talk or in the breaks. So what does DNMT3A do? How does it cause TBRS? What can be done? I don't know all the answers to all these questions, but we will address some of this. Okay, so all of you came here from afar, some of you from very far. I know somebody's here from Malta, and some came from California, and some came from Texas. And whether you flew or took a boat or whatever, at some point you had to drive to get here. And so here we have a map of the roads in the United States, and there's lots of roads, and they all go to different places, and these are most of the big roads. And then you got to Texas, and then there's more roads in Texas, and these are some of the main roads. There's San Antonio, kind of right in the middle here. And you can see Houston, where I'm from, is over on the right here. And you can even drill down, and here's San Antonio, and we're up here somewhere on the right. So in order to go on these roads, you need to have good quality roads. And hopefully, when you drove on the roads, they were good quality roads, sort of like the ones that I show here. There's a highway here or a nice quality road out in the middle of nowhere. But these are good roads. Why are they good roads? The pavement is clear, there's good markings, the boundaries of the roads are clear, so you know where to drive on the roads. Hopefully, you weren't driving on a road that looked like this, although there's clear boundaries because those trees are making clear boundaries, but it doesn't look like a very good road, and if you're going down that road, you're not going very fast. And here's another road, and really, I hope you weren't there. And if you were on this kind of a road and there were cows in the middle of the road, you might think, I'm not even in the right place at all, okay? So good roads are really important for us to get to where we want to go quickly. Good roads need great equipment. You've got to have equipment there to make those good roads, and we all know that roads are actually incredibly expensive to make and maintain. So I want to pose to you that DNMT3A is kind of like one of these massive pieces of equipment that is necessary for the roads, okay? So, I'm trying to remember which slide I have next. Yes, okay. So before I go to the next slide, so DNMT3A is actually a big protein, okay? We talk about genes, you hear about mutations in genes, but the gene really codes for making a protein, and a protein is just a thing in the cell that has to do something, okay? And one way to think of DNMT3A is that it's involved in making the roads. And so I bought this really nice looking truck, and hopefully some of the kids can play with it later. And what you can see is this is a pretty fancy piece of equipment. It's got all this movement here, it's got this shovel, it's got the excavator piece, it's got a cab, it's got all these wheels, it's got little stabilizers here. I mean, this is a multi-featured protein, right? It does a lot of stuff. And DNMT3A is like this. It's a multi-featured protein. It's big. It's about 1,000 pieces that put the whole thing together, and we can name them all one by one. And when we know what your mutation is of your child, it's one of those 1,000 pieces that is basically broken on this major machine, okay? So DNMT3A doesn't do this all by itself. It does have friends. And just like when you're building the road, you have to have one of these really cool things that makes the pavement really good, and you have to have your cement mixer and another thing, and you have dump trucks, and all of these things are important together to make sure that DNMT3A can do its job. And they have to work together, and part of the way they work together is by communicating with each other, and sometimes they physically interlock in different ways inside the cell, and all of this makes the road construction work really well. So, a major, can't see my own slides. So a major role of DNMT3A is to build the roads. Sorry, I got to get rid of this so I can see what I'm doing here. Okay. A major role of DNMT3A is to build and maintain these DNA highways. So why are the DNA highways important? It's so that the rest of genes can be turned on and off and regulated properly. And I'll come to that a little bit more in a minute. So why are the good roads really important? And so then you can come back to the analogy of the United States. So we have all these regions in the United States, different cities and different regions. And there's actually overlap in what all these different cities do. But they kind of are specialized, right, in certain functions in our country. So you might think of California for a bunch of different things, but we know there's Silicon Valley. That's where a lot of computational power has been developed. Houston and Dallas are kind of the oil capitals of the country. There's a lot of farming that goes on everywhere, but largely in the Midwest. Manufacturing on the east, everywhere, but also particularly in the Midwest, the East Coast. Boston, education, technology, government. Sometimes we think we could get rid of that altogether because it's being pretty messed up these days. But all of these are together what make the country work. And part of what makes it work is to have a good communication system. Those roads, those airports, all of that is part of this communication system to make a good quality functioning country, mostly functioning most of the time. And DNMT3A, again, is building these roads, maintaining those roads, making sure the pavement is clear. Even if you think about airports, DNMT3A, these devices are also in airports, making sure the runway is there. And the borders, again, the borders of each of the runways is clear so the planes can land when they're coming down. So DNMT3A is really critical for doing all that. So if you take this analogy of these cities, you have different genes doing different things in different tissues. Some groups of genes are running the brain, and some groups of genes are running the heart, and some groups of genes are running the bones and the bone marrow itself and the blood system. And all of those things have to work together. And again, DNMT3A is important for making sure that they are established well and that they can communicate with each other. So the cities are like the genes or the groups of genes with a common function, and they need to communicate to allow the country to work together. So we've seen this map now a few times. So how can we now relate this map of DNMT3A and the mutations in DNMT3A to what I've been telling you about DNMT3A and what it does? Well, you've seen in these maps these sections of the gene, these domains, and we call them the PWWP domain, the ADD, the MTAse, the methyltransferase domain. And you can really think of each of them as different chunks of this giant protein. So in a way, the most important part of DNMT3A is this methyltransferase domain. It's really the business end. It's putting these methylation pieces that you might have heard about onto the DNA, and that's important for making and allowing the DNA to communicate properly. And so you can imagine if you have a mutation here in this piece, that's going to cause some problems in the road construction. But similarly, you can have a mutation here in the ADD domain, and so it's a mutation down here instead. And in fact, one of the most important regions is this small region here, the PWP domain, and the reason I picked the little cab is because it is helping to direct the activities of this protein to places. You can imagine if you just had the little digger and there was no controller here, then it would be going all over. It might be building roads when it shouldn't be, or it might be putting dirt on the roads instead. And so all of these other pieces are important for controlling the activity of that business end of the protein. So now you can have little pieces missing on any part of that protein, and that can affect the function of DNMT3A. Okay, so we might ask, do mutations all in the same domains have similar impact in patients? This is something that the organization is trying to address, to understand, partly to be able to predict the future and understand if you're diagnosed with a certain mutation, what are the other likely problems you're going to have. It would be nice if we could say, well, if you have mutations here, there's going to be learning disabilities. If you have mutations here, there's going to be heart, and so on and so forth. It's really not that simple. We wish that it would, but partly we're trying to collect all the data to understand that. If the function, you can imagine if there's any piece of this that's really damaged badly, you're probably going to have a similar outcome and really a non-functional protein that's not going to be able to participate in the road building. But there's huge variability, as we know, and we don't know if this is because there's other genes in the individual that contribute to that, or whether it's really the position of the mutation. As I told you, there's 1,000 pieces that are needed to put this together, and any one of them, or many of them, can contribute to losing the function of DNMT3A. So we think the family registry or the natural history that was talked about this morning is going to be important for kind of correlating what the impact is on an individual and where the mutations are in different parts of the protein. So, okay, what else can we learn? This is the model. So one of the things that we're trying to do in my lab, we're a basic research lab, is model some of these variants in the mouse. And what we would like to do is make some models with mutations in this part and some models with mutations this part and this part, because then we can compare them. And mice are kind of easier to compare because otherwise they're basically exactly the same. So this is another map, and these are some of the mouse models that we're working with in our lab. You see 3 different mutations here, and then one is a loss of function where we delete the entire function of the gene. So some of this, some of you may have seen before. I think Ayala Tovi, one of our colleagues, presented this before. But we made one mouse with this 293 mutation, and as you can see just at a glance, these mice get very big. This is about 6-month-old mice. You can see on the bottom they're a lot bigger than their sibling on the top. And we can measure the weights with many mice over time, and you can see the TBRS modeled mice in red, they get heavier very quickly over time, whereas the other ones just gain weight like regular mice. So there's a lot more data on this, and we found that these particular mice recapitulate a number of the phenotypes of TBRS patients, but not all of them, they are mice. So then we decided to study this particular mutation more, this 293 mutation, and understand it's in that CAB domain, right? It's in this PWP domain, this controlling domain. So what else could we understand about that and how it affects things? So yes, there's the CAB of the protein right there. So what we realized by studying just this one is that there's this major quality control system in the cell, and that when we had this mutation in the 293, what it did is this quality control mechanism in the cell recognized the mutant protein with the broken CAB and took it straight to the junkyard, okay? So we can call this a junkyard mutation because it's recognized very efficiently and sent to the junkyard. So what's the significance of that? Well, the junkyard, it's amazing, you know, we have great junkyards all around the country, and you have them in your cell too, and they're really, really important for getting rid of proteins that are non-functional for a variety of reasons, even in older cells and older individuals, and recycling the materials, right? There's lots of good metal in there, there's tires, there's all sorts of stuff. And here's some nice pieces of a junkyard, and this whole system basically recognizes them, sends them to the junkyard, recycles them. So we wondered, these 293 mutant cells, they have this, they get sent to the junkyard. So are there other mutations in TBRS that also are kind of a junkyard mutant so that they're automatically sent there? So it turns out that DNMT3A mutations, we know, basically define TBRS syndrome, but there are also a lot of older individuals who you can find these mutations in their blood. And some of you will be familiar with the term clonal hematopoiesis. It causes some confusion in the TBRS field, but basically most older individuals in the blood will acquire some DNMT3A mutations, and they're mostly perfectly harmless, but they allow us to study what happens with the TBRS-related mutations. So here we have lots and lots of mutations because there's lots of elderly people and we can identify these mutations. There's many more that carry them than there are TBRS patients. So we decided to map how many of these from either TBRS patients or older individuals were basically these junkyard mutations. And I won't go through this little map that we did here, but we've tested whether these individual variants were sent to the junkyard or were stable and could still be used in the cell in some degree. And what you can see is that anything below the little dashed line here in red, those were the junkyard mutants, and all of the ones at the top were stable. It doesn't mean that they're perfectly functional. They just don't get chewed up by the junkyard system, but they may still be broken in some way. And you can imagine having a broken, digger can be just as problematic, and sometimes you might think you'd rather get rid of it altogether than have one that causes other problems. So it doesn't mean that they're normal by any means. It just means that they're not sent to the junkyard. But we found that about a third of the mutants get sent to the junkyard. And we could classify those together. Anything that gets sent to the junkyard, a third of those, sort of behaves similarly in the blood, okay? They just disappear completely. You still have some DNMT3A protein around because you have two copies and there's enough made from the other allele, the other gene, but the one that has the mutation, those are made into the protein and sent to the junkyard. And those all seem to behave similarly. So now in my lab, we're studying the way the junkyard processes DNMT3A so that we can modulate it. So we're hoping that if we can actually slow down the junkyard process, that recognition or the recycling process, that we might be able to boost the amount of DNMT3A in the cell and thereby potentially have some therapeutic effect. Okay, so by studying just this one mutation in this one patient, we discovered the process of recycling through the junkyard, and now we have new ideas about how to change that and potentially affect not just that 293 mutation, but it could potentially affect the other 1/3 of mutations that also get sent to the junkyard. Okay, so we're studying the way that, whoops, I went backwards. So we think this junkyard system, the junkyard mutations, will be relevant to other mutations, but not all of them. It's taught us about what controls the stability of DNMT3A in these cells, and as I said, we're trying to modify it. So to sort of summarize what I've told you before we go on to Rachel's talk, and she'll return to some more clinical aspects, DNMT3A is really this multifunctional piece of equipment. It's important for facilitating that communication between these other groups of genes in the cell, those other cities. And we think that either by studying one mutation at a time, as I showed you with this 293 mutation, or many mutations, as I showed you in that graph where we studied 250 mutations in a very high throughput assay for whether they get recycled or not, that it can give insights that will be applicable more broadly to hopefully many other patient mutations. So I will leave it there, and hopefully the analogy was useful, and I'm happy to talk more about the technical aspects later. I think we'll switch to my colleague Rachel, and then we'll come back and we'll both take questions together. Okay? Okay.
Hi, thank you. I feel like I'm this giant floating head on the screen, but I'm really excited to be here. Sorry I couldn't be there in person. Certainly, it's such a great meeting, and I've been participating online, and it's always just incredibly motivating to continue the work we do. when we hear great science and then we hear about the building of the registry and other exciting initiatives. I feel like I needed a toy. It has nothing to do with DNMT3A, but go Astros. I felt left out with Peggy's trucks. But today, as Peggy talked about, I'm going to talk about the impact of DNMT3A mutations on blood development and leukemia risk. I'm a pediatric hematologist oncologist, which means I specialize in the care of children with blood disorders, including leukemia. And so DNMT3A mutations have become a real focus of my work, largely revolving around understanding blood development in individuals with TBRS. So as a pediatric hematologist oncologist, I get asked questions from the TBRS community, and this is just a smattering of sort of the ones I find most frequently being asked. Are there blood abnormalities associated with TBRS? Are individuals with TBRS get at increased risk for blood cancers called leukemias? And if so, are there appropriate monitoring or surveillance that should be done? And then if leukemia does develop, are there specific treatment for it? So I'm going to work my way through the answers to these questions, and I'll start by saying I don't have all the answers to these questions, but I think they're good ones and ones we all need to continually think about as we study this particular disorder. So just to start with how blood develops normally, to get a sort of basal understanding of how this process works. This is a process that happens in the bone marrow, so that hollow center of the bone is where all the blood cells in the body arise from. And the process of making blood cells is this hierarchical process that begins with a specialized cell called a blood stem cell. And a blood stem cell is really special in that it can make new copies of itself to maintain that stem cell pool, but it also can differentiate, meaning it can give rise to all of the mature blood cells that we need in our bloodstream to support our health. And we have a lot of different blood cells that we need floating around in our blood to maintain function, to maintain our function. The erythrocytes, or red blood cells, are the component of the blood that carries oxygen from our lungs to every tissue in our body, to the brain, to the muscles, and all places in our body that need oxygen delivered to them to function. Blood platelets are important in making sure that we aren't prone to bleeding. So if you get a cut, the platelets are the things that plug up the hole to prevent you from continuously bleeding from that injury. The rest of the cells are all together called white blood cells, and these are really our infection-fighting system. So they're responsible for preventing serious illnesses from invaders like bacteria and viruses. Now, of course, that's when things function normally, but abnormal things can happen during blood development as well that lead to a number of different problems. If you don't have enough red blood cells, for instance, that causes a syndrome called anemia, where you might be pale and tired. If there are issues with the platelets, it might increase your risk for severe bleeding. Conversely, if the platelets are kind of too active, then you might be at risk for blood clots forming throughout the body. The immune system is obviously very important, and things can go wrong with it. If it doesn't work properly, then you could be at risk for having more infections than other people. Conversely, it can actually be hyperactive, and that can cause annoying things like allergies, or very serious things like autoimmune issues, lupus, things like that. So all these things need to be quite precisely coordinated and orchestrated over time to make sure that the blood remains in its normal functional state. And of course, the sort of worst case scenario when things go wrong with the blood is the development of a blood cancer termed leukemia. And how that happens is essentially bad things happen to some of these cells in their early stages of development. And I intentionally have multiple lightning strikes here because usually just one bad thing happening to a cell isn't enough to make it into a blood cancer. It usually takes lightning striking multiple times in the same cell to become a blood cancer, which is why these are pretty rare events, because it takes these multiple lightning strikes to ultimately converge to develop a blood cancer. And why do we have this concern about individuals with DNMT3A mutations? Well, it's because for some years we've known that individuals with blood cancer, if you look at the cells from their cancer, relatively high frequencies of these patients will have DNMT3A mutation in their cancer cells. Now, I will point out that that's pretty much restricted to older adults with blood cancers. Because you can see in acute myeloid leukemia, one of the most common adult cancers, about a quarter of individuals will have DNMT3A mutations. On the other hand, if you look in children, with the three most common types of blood cancers we see in children, almost none of them have DNMT3A mutation. And again, I think this talks to the fact that it takes multiple events happening to the same cell, usually taking years of time to happen, for a cell to go from a normal blood cell into a cancer blood cell. Additionally, just as Peggy mentioned, there are lots of people walking around with DNMT3 mutations in their blood who are otherwise healthy. In fact, if you sequenced anyone age 50 and older deeply enough, if you looked at enough of their blood cells, in a vast majority of them, you would find DNMT3A mutations at even low levels. But in some individuals, those DNMT3A mutations will be more present in the blood, and you can readily detect them with sort of more standard laboratory assays, and that's the process called clonal hematopoiesis, where you have a blood cell clone that's preferentially expanded and is more represented in those blood cells that we see floating around in our system. Even these individuals, while they are at a slightly increased risk for the development of blood cancers compared to individuals without that clonal hematopoiesis, a vast majority of them will never have cancer. They'll just live the rest of their lives with these DNMT3 mutations present without ever having a real adverse event because of it. So I think that's, again, sort of pointing to the fact that it takes multiple lightning strikes to have a blood cancer. And lightning strikes aren't that common in the same cell. So the cancer risk, even when you have DNMT3 mutations present, isn't exorbitantly high. Now, all of the things that we've talked about so far are talking about individuals with DNMT3 mutations just in their blood cells. Of course, individuals with TBRS essentially have DNMT3 mutations in every cell in their body. When we first started learning about TBRS, we didn't really understand if there were blood phenotypes associated with it, changes in the blood development in individuals with TBRS. So we set about to answer that question. To do so, we really leaned on the TBRS community, and Kit's presentation today, I think, really summarizes how we feel. This is a real partnership between scientists who are thinking about these disorders in the lab and all of you out there in the community. And with that participation, we were able to collect blood samples from individuals really from around the world. And so it was a great partnership. We did really deep analyses of those blood samples that we got from 18 individuals with TBRS, aged 2 to 38 years of age, and then age-matched individuals without TBRS. So most of them were the siblings of those with TBRS. So thank you to those families who have had their brothers and sisters donate blood as well. And then 4 unrelated individuals who were matched for age. So just sort of walking through the results of that study, what we found is that when we looked at the different types of blood cells, we saw some consistent and very subtle abnormalities. So if you just look at those red blood cells, essentially red blood cells, there's a range of normal size of red blood cells. If you look at 100 individuals, you'll see some that have relatively smaller blood cells, some that have relatively larger blood cells, and then sort of a spectrum in between. When we looked at the individuals with TBRS, we found that on average, the size of their red blood cells was slightly larger than our unaffected control individuals. Most of these still fell within that spectrum of the range of normal. And whereas the siblings sort of sat right smack in the middle of that range of normal, our TBRS individuals tended to shift towards the higher end of that range of normal, with just a few sort of just outside that normal range. This was not associated with any evidence of anemia, so the number of red blood cells was normal. It was just this pretty subtle difference in the red blood cell size compared to the unaffected siblings. So the summary of the red blood cell findings is that TBRS individuals tend to have larger red blood cells. It is not associated with anemia, so it seems to be a benign condition, similar to what's seen in another genetic disorder called Down syndrome. Down syndrome individuals tend to have larger red blood cells than non-Down syndrome individuals. And we think this is, again, most likely a benign condition. And so this is a point that I'll make. It's important to sort of know what your normal is. So I think if you have a CBC, a complete blood cell count, which is a lab test that would give this particular information, at a time when you're perfectly healthy, it would tell you sort of what your normal red blood cell size is. And that's helpful because if you have an illness or something and you have a blood count drawn and the doctor sees that your red blood cells are slightly larger, it might lead to an unnecessary number of evaluations to look into that. But if you know that's just your normal situation, then it could avoid some of those unnecessary workups. I'm not going to talk about the platelets because we actually didn't see any consistent differences in the platelets, either in number or in size or other features, when we compared our TBRS individuals to the unaffected individuals in our study. But when we looked at the white blood cell populations in our individual patients, we did see some significant differences. So one type of white blood cell in our infection-fighting armamentarium are called blood neutrophils. And we saw that when we looked at our TBRS individuals, they tended to have more blood neutrophils than our unaffected control individuals. And neutrophils, again, are part of our sort of primitive infection-fighting system. What neutrophils do is when they sense danger, usually it's from something that has caused some level of inflammation, that signals the neutrophils to come into that area and help fight it off by kind of consuming and destroying the thing that's causing the inflammation. It also releases somewhat toxic granules from its inside, and those are factors that help to recruit more neutrophils, but can cause some local inflammation at the site as well. What we wonder about is in TBRS, are the numbers of neutrophils elevated because of other sources of inflammation? And we've heard about obesity in a number of the talks yesterday and today, but obesity is also a source of inflammation, and we're wondering if that might be driving this increase in neutrophils, but we don't have all the answers quite yet. But we do wonder if it's a result of increased basal inflammation in individuals with TBRS. The next white blood cell type that we saw different amongst our TBRS individuals were the so-called blood B cells. And we saw that these were significantly reduced in our TBRS individuals compared to the unaffected controls. And blood B cells are a slightly more sophisticated part of our immune system compared to the neutrophils. Our B cells, when exposed to something like a vaccination or an infection, they sort of scan that to see if it's self or a foreign protein. Once it identifies it as a foreign protein, then it turns into this factory that produces infection-fighting proteins called antibodies. So that's effective at sort of clearing the precise insult that the B cell has scanned for and identified. At the same time, it makes what are called memory B cells. So these are cells that are sort of like poised so that the next time the body encounters that same agent, it's just ready to mount an attack very quickly, which is why vaccinations are quite effective. So with the reduced numbers of B cells that we see in our TBRS individuals, it possibly means that these individuals are at baseline making less antibodies compared to non-TBRS individuals. It could also result in suboptimal responses to vaccines, although we don't know if that's the case or not. And potentially, we have heard about some increased susceptibilities to infection, so perhaps this is one contributing factor in some individuals with TBRS. But all things are actively being explored right now. So to just summarize what we found in the individuals with TBRS compared to the unaffected siblings, we saw that the red blood cells tend to be larger in size on average. We think this is probably benign and requires no intervention, but again, the importance of knowing your normal. Amongst the white blood cells, we see that increase in neutrophils, which perhaps is a reflection of basal increased inflammation. So maybe some of the measures we've talked about for weight control might help to combat that issue that we see in blood development. And then I didn't talk about another blood type called the T cell, and we saw mild subtle differences there, but the fewer numbers of B cells, could that lead to an increased susceptibility to infection in TBRS individuals? So things that could be considered are checking what are called vaccine titers to make sure an appropriate response has been mounted when you've received a vaccination. It might mean you need a booster slightly earlier than other individuals do for some things, for instance. And then perhaps in individuals who are really battling with recurrent infections, it might be wise to check immunoglobulin levels. There are basic lab tests that can be done to look for that. Okay, so I think one of the most pressing questions amongst the TBRS community are individuals with TBRS at risk for leukemia? We've seen a smattering of reports in the literature, and Dr. Tatton-Brown yesterday talked about the number of individuals in the registry who have reported cancers, including blood cancers. And so far, it seems, maybe 8 or 9 individuals with TBRS have reported some type of blood cancer. So let's say that that's 8 total. I think we still can't yet quantify the risk of leukemia at this time. Number one, we don't know if 8 is the absolute number. There are probably individuals out there who have had a leukemia, but we haven't identified them yet. We also don't know the denominator. We still don't know how many individuals with TBRS there are in the world. So it's hard to quantify the absolute risk of developing leukemia amongst TBRS individuals. I would say it's too early to say what is that absolute risk. One of the ways we can sort of start to maybe address it while we're still gathering data from the TBRS community is to think about the use of these mouse models that you've heard a lot of talk about yesterday and today. And I think there are a number out there that can be used. Peggy just talked about a few that we've used in our lab. There's another that's been used at WashU and in other labs that are really useful models to study all the phenotypes of TBRS because we know they do recapitulate some of the basic sort of almost universal features of TBRS, so namely the overgrowth and some of the behavior and developmental issues that we see are mirrored in mice, at least to the degree we're able to study them. So how did our mouse models compare to our humans in terms of the blood development issues that we saw? So if you just look at red cell size, it was somewhat gratifying to see that this is also recapitulated in all 3 of the mouse models we looked at. Those mice had larger red blood cells than their wild type siblings. We also saw that same phenotype of increased blood neutrophils in our mouse models and decreased blood B cells. So we think not only are these good models for the overgrowth and perhaps the intellectual and behavioral issues, but they seem to be really good models for studying the blood phenotypes and TBRS as well. So we can use these mice to generate larger numbers and follow the mice over time to maybe get a slightly better handle on what's the absolute risk of cancer in individuals with TBRS, although these aren't perfect experiments, of course. And what we saw is that we do see that cancers are slightly more common in mice with DNMT3A mutations compared to their what we call wild type siblings. We've talked about a number of different TBRS models, and the incidence of cancer in those models has varied quite substantially. I think it's problematic to compare them one-to-one, however, because we haven't yet taken them all in the same place, made at the same time, and followed them together. So I think it's hard to say, based on the existing data, if there are clear differences based on the type of mutation that we have in the mouse model yet, but those experiments need to be done. But what I want to emphasize is that overall, the vast majority of mice that have been studied have not developed cancer in their lifetime. They've lived an entire normal lifespan of a mouse without ever developing cancer. And when the cancer developed, it took a very long time for it to do so. Again, lightning needs to strike multiple times to have cancer, and even in the mouse models we've seen that, and that it only develops in a fraction of the mice and only once they've reached a certain age. So this is a heavy topic and a difficult one to discuss, but what I'd like to emphasize is it seems, based on the data we have available, cancer is not an inevitable consequence of having a DNMT3A mutation. It seems that there may be a slightly heightened risk, but I believe a majority of individuals with TBRS will not develop cancer in their lifetime. So what are our current recommendations? Well, that's tricky. I think as everyone here has mentioned in some way, shape, or form, we need to collect more data, and we're doing a really good job of that. The registry is amazing, and it needs to be continually have more data added to it, including with incidence of cancer, including blood cancers. But as Dr. Tatton-Brown talked about in her talk yesterday, there are probably other cancers we need to get more information about to see if there's really an association or not. And then, of course, looking at other blood and immune system disorders is going to be really important to better understand those consequences of having a germline DNMT3A mutation. And again, I'll just put in a plug-in for continuing to put your data into this amazing registry that's really been already a source of great data for research, but will continue to do so as it builds. Okay, so monitoring is a common question. And what I have to say is even doctors haven't reached an agreement. I think it's a really tricky subject, and I know Dr. Tatton-Brown talked about this a little bit yesterday. It's hard to know. If we haven't yet nailed down what the incidence is, it's hard to say that it needs to be screened for. Additionally, there aren't really great screening tests for leukemia right now. With the current technology we have, there's not one test where you can say, oh, this is definitely going to be able to identify somebody who's at risk to develop a blood cancer. It just doesn't exist. Maybe as technology evolves, there will be better ways to detect and look for it early, but at the present time, that's not really possible. Additionally, unlike breast cancer, where if you catch it early, there's a different treatment, a different outcome, that doesn't exist for leukemia right now. There's no early treatment. So if you do happen to pick it up before symptoms develop, the treatment would be the same. The outcomes are the same. We know this from large studies of patients who have developed leukemia. So with the current treatments available for leukemia, there's no early treatment. So right now, at least the blood cancers sort of fail the two major sort of boxes to tick in terms of something that has a really great track record for surveillance. There's not a great screening test to detect it, and even if detected early, there's not a treatment for it. So at this time, based on the current technology we have available, the current treatments we have available, I think the most important thing for families to know are possible warning signs for leukemia. So you could go to your doctor if you have concerns based on symptoms that have developed. So just very quickly to review, what are the signs and symptoms of possible blood cancers? So as I said, blood is made in the bone marrow, and because that's where blood is made, that's often where leukemia arises as well. And what happens is those leukemia cells grow much faster than our normal blood cells. So they sort of crowd out the normal blood cells over time, making it hard for the body to produce enough of them. So that leads to a lack of red blood cells, which makes you anemic. So you have pale skin, you get really tired, you might get short of breath much more easily than you normally do. If you aren't able to make enough platelets, then you might be at risk for bleeding. So perhaps bleeding gums when you brush your teeth, really bad nosebleeds that happen often and are hard to stop. And really easy bruising. So bruises in places that you can't really explain. Like we all bang our shins and get bruises, but if it's in place you're like, I can't imagine that I banged that on anything. Or if it's bruised, that's really, you can feel it. It's not just level to the skin. Those might be warning signs that your platelets aren't quite what they should be. If you don't have enough white blood cells, you're prone to infections, so patients can present with persistent fevers. And then because all these cells are made in this really confined cavity inside the bone marrow, the mere expansion of those leukemia cells over time can result in some persistent bone pain, which is one of the common presenting features of leukemia. Okay, an important question, can leukemia be cured? Yes. Most children diagnosed with leukemia can be cured. And so I want to emphasize that even if leukemia is detected, we have measures available, not just to treat it, but to cure it. And those treatments vary and depends on the type of leukemia that it might be. All patients with leukemia get some form of chemotherapy, but how much and exactly which ones, it depends on the type of leukemia that has developed. There's a lot of talk about immunotherapy. It's essentially treatments that harness the patient's own immune system to help fight off these blood cancers, and really exciting data, and they're increasing in use, more so in some cancers than in others, but it might be a possibility. And then lastly, bone marrow transplant is something that I'm often asked about. So this is only used for certain cases, but this is a procedure in which a patient is given high doses of chemotherapy, sometimes radiation therapy as well, with the goal of eliminating all the blood cells from the patient's bone marrow, both good and bad blood cells, and then they're replaced with blood stem cells from a donor. So just a plug, that I would encourage friends and families to enter themselves in the Be the Match program. And you just usually send in a buccal swab, and then that puts you in this pool of individuals who could potentially serve as a bone marrow donor. So I'll wrap it up there by saying that amongst our TBRS individuals and in our mouse models, we've seen very subtle and somewhat variable differences in blood development in TBRS. Know you're normal, just it might avoid some unnecessary workup down the road. Cancer is uncommon. There aren't good screening or surveillance things right now, but it is important to recognize the warning signs that would lead you to medical attention. If leukemia occurs, cure is possible. And lastly, I'll just say it again. Please participate and continually participate in the TBRS registry. It will allow us really powerful data and information with which to answer some of these many unresolved ongoing questions. With that, I'll stop. And if questions arise that you'd rather ask me later offline, there's my contact information. I'd be happy to chat.